Before I begin, I would like to extend my sincere gratitude to the program director for the opportunity to present my research results today. Protocol observed. Good morning, good afternoon, good evening to everyone. My name is Mahlodi Maripa. I am currently enrolled for PhD in Agronomy and Soil Science at the Department of Crop Science at Tshwane University of Technology. I am supervised by Prof. Felix Dakora and co-supervised by Dr. Titus. My talk for the day is selecting 27 field-grown common bean genotypes for symbiotic performance and drought tolerance at Malken's Research Station, Eswatini, which is formerly known as Swaziland. This is my presentation outline where I'll be talking about background and introduction of common bean up until conclusion and recommendations. What is this common bean? Common bean is a legume grown for its edible seeds. It is a food in many countries, including sub-Saharan Africa. Common bean has many benefits. Nutritional benefits, it is rich in protein, fiber, micro and macronutrients, and carbohydrates. Those who don't know common bean, there is the picture of common bean. It also has environmental benefits whereby it improves soil fertility by fixing atmospheric nitrogen. Remember, we have around 78% of nitrogen in the atmosphere. However, it cannot be used by anyone. Therefore, planting these legume crops, they end up fixing the nitrogen. By doing so, they also improve the N economy in cropping systems and adding nitrogen to the soil. But then we have production constraints of common bean, whereby it is low soil fertility, pest and disease, and erratic rainfall. Let's look at this table. This table is showing the production of common beans at Eswatini from year 2016 to year 2023. In 2016, people at Swaziland, the community members and the farmers, they were only able to produce around 700 metric tons of common bean. However, the total consumption annually, it was around 3,654.5. You can see the gap. Again, in 2021, they were able to produce around 659.6, and their total consumption was around 5,000. And in 2023, the production reduced more by 582.5, and their total consumption was around 7,370. You can imagine the gap. So with the research gap again, we have realized that these people from Eswatini, they are heavily relying on imports from countries such as South Africa and Mozambique. Therefore, now, when I was sitting with the community members and the farmers, because we have like collaborations that side, they were telling me that they want this common bean production because now they're using it for many purposes. Some people, they were saying they're using it for medicinal purposes. Others, they were saying they're feeding it to their livestock. Other people, they were saying it is stable food for them. They eat it almost every day. And the other ones, they were saying they're selling this common bean on the street so that they can support and provide for their families. Therefore, that's when now I found that this research is impactful, because now when you are doing research, you really need to bring solutions to your community and to your nation. It's not about being a doctor or being a professor, I'm sorry, but then it's about bringing the solutions to the people that are in need. These are my objectives. I was determining plant growth, symbiotic and performance, grain yield of 27 field-grown common bean genotypes. And again, I've measured the photosynthetic water use using the infrared analyzer and the carbon isotope discrimination techniques. Methodology. My study site was at Malken's Research Station, Eswatini, during 2023 to 2024 cropping season. And then the experiment was designed in the randomized complete block design, whereby we have replicated 4 times. And then again, that is the layout. of my research, and then you can see the intra and intra-row spacing that I have used for my research. And then down there is Malkin's research station. And then the second picture, it is common bean when it was still growing. The third picture is showing common bean during podding stage. Plant sampling and processing. At the early pod filling stage, 5 plants were randomly harvested from each plot. Shoots and nodulated roots were separated and oven dried. The shoots were weighed and grinded to a fine powder for the analysis of 15N and 13C. This is a picture of the pods, and then next picture is the samples that I have prepared to go to isotope laboratory at Stellenbosch, Cape Town, South Africa. These are my 2 equations that I have used to calculate delta 15N isotope and delta 13C isotope. I then further measured gas exchange, whereby the parameters that we focused on was photosynthetic rate, stomatal conductance, intercellular carbon concentration, transpiration rate. And then the measurement, we're taking them early in the morning, around 8 a.m. to 10 o'clock when the sun was out. And then the equipment that was used was portable infrared gas analyzer version 6.2. With the statistical analysis, all data collected was subjected to analysis of variance ANOVA using statistical software version 10.1, where significance difference were observed. Then Duncan multiple range test was used to separate the means. Results and discussion. The results and discussion of those 27 common bean genotypes. With this table, you can see that we have managed to check the symbiotic performance and plant growth of those 27 common bean genotypes, whereby we have seen that the genotypes that recorded the highest shoot dry matter also recorded the highest nitrogen content, and then they further continue to record the highest amount of nitrogen fixed, and then again they recorded again the highest amount of soil and uptake. 11 out of 27 common bean genotypes showed the potential to increase the grain yield. And then the genotypes that recorded the lowest shoot dry matter also recorded the lowest nitrogen content, and they further recorded the lowest amount of nitrogen fixed, and they again recorded the lowest soil and uptake. And then these genotypes that recorded the lowest shoot dry matter, they showed that their grain yield was also very low. And then again, we have seen that the genotypes that recorded the lowest delta 15N, they have recorded the highest nitrogen derived from atmosphere. Continue with gas exchange studies. On this table, we have realized that the genotypes that recorded the highest shoot dry matter, they also recorded the highest carbon content, and the genotypes that recorded the lowest shoot dry matter, they recorded the lowest carbon content. And now continuing, we have observed that the genotypes that recorded the highest photosynthetic rates, they also recorded the highest stomatal conductance, and then the genotypes that recorded the lowest stomatal conductance, they recorded the highest water use efficiency. And amongst 27 common bean genotypes, only 4 showed the potential to be regarded as drought-tolerant cultivars because they were having less amount of or less negative amount of 13C, whereby it was telling us it is high water use efficiency. With the discussion, it is just the recap from those tables, whereby we have seen that 3 of the genotypes exhibited superior shoot biomass, and then 11 out of those 27 common bean genotypes recorded relatively much higher grain yield. And then the genotype that recorded the highest grain yield, it was RM4. Continue again with water use efficiency. The delta 13C Less negative, it stated that the less negative values means high water use efficiency, and high negative means reduced water use efficiency. That's when we have identified that for common bean genotypes, they can be regarded as drought-tolerant cultivars. And then again, we have also seen that genotypes with high photosynthetic rates exhibited larger stomatal conductance, and then the genotype with low stomatal conductance resulted in an increase in water use efficiency. And then continuing with 15 N values and N2 fixation, we have also realized that 11 genotypes derived their nitrogen from the atmosphere by 50% and more. Genotypes that have recorded lower delta 15 N values derived most of their nitrogen from the atmosphere. This is just the recap from that table. And then the genotypes which recorded the highest shoot biomass also recorded the highest carbon content. With the conclusion, the results, the study revealed significant variation among genotypes in terms of growth performance, nitrogen fixation, carbon accumulation, grain yield, etc. And this genotype with superior performance also showed or recorded increase in shoot biomass, grain yield, and fixed and water use efficiency. And we also identified that there were two genotypes that were able to derive more of their nitrogen from the atmosphere. And again, we also identified that there were 4 genotypes that showed the potential of being drought tolerant. And then this study confirms that strategic genotype selection can contribute meaningfully to closing Eswatini bean production gap while improving soil health. In recommendation, I would like to say, genotype with high superior performance need to further be studied and tested. And then again, support extension are needed for increasing local common bean production to reduce import dependency and enhance national food security. And my last recommendation is that future studies should explore genotype together with the environment interaction, particularly under drought stress, to strengthen climate resilience strategies. I would like to acknowledge Prof. Felix Dakora, which is my supervisor, together with Dr. Titus, Tshwane University of Technology, that's where I've enrolled my PhD, Science Forum, and MDP, for giving me the platform to share my research results, National Research Foundation, for funding my studies. Thank you so much. And I would like to have more collaboration based on my study and more networking, and I'm open for opportunities, and I'm also open for postdoctoral opportunities in any country. Thank you so much.